Hey, what is up? It is Saturday, March 26th, 2022. We are hitting the end of March today. No, we're not quite yet, but we're getting close. We've definitely passed spring, the start of spring, or the solstice or whatnot. I don't know how all that really works, even after all these years. It wasn't the solstice, it was the equinox, I guess. Whatever. Anyway, so we're into spring, the flowers are blooming, the grain is growing, the birds are chirping, and rabbits are, doing the rabbit thing, right? Basically, they're all in heat. Anyway, so with that being said, let's try to finish up Metabolism March strong. Like I said yesterday, I don't think we're going to do that, but we will get through a significant portion of metabolism, the metabolic cycle, and today is an interesting day for me personally because I'm finally able to link metabolism through my studies to mTOR. Now, mTOR is implicated in a lot of anti-aging discussions, and for those of you who do not know, we don't want to get too deep into that right now, but I'll make that connection later on towards the end of this video. But for now, let's just talk about, we want to talk about ATP, AMP regulation, glucose glycolysis regulation in specifically right now skeletal muscle cells, but in general. In skeletal muscle cells, the processes that I'm going to be talking about are most prominent or prevalent because skeletal muscle cells, they require the most ATP since they are the so-called workhorses of the body, right? So with that being said, let's talk about glycolysis. So you remember, we're going back to glycolysis. We always have to go back to glycolysis because glycolysis is how we get ATP and initiate other ATP getting processes, right? Like the Krebs cycle, the transition phase, the Krebs cycle, or citric acid cycle, etc., and then the electron transport chain, which then leads to ultimately the most significant portion of ATP in the body that we can get. Glycolysis also produces ATP as well. So speaking of the ATP production of glycolysis, why do I have phosphofructose kinase up here? So if you recall, phosphofructose kinase is the enzyme which is involved in the third step of glycolysis, right? Phosphofructose kinase right here converts fructose 6-phosphate to fructose 1,6-bisphosphate. Okay, remember at this point, fructose 1,6-bisphosphate. At this point, this is called the committed step of glycolysis, or the most highly committed step, and thus this is the most highly regulated step of glycolysis. Because once we get here, there's really no going back from this point. Fructose 1,6-bisphosphate has to go through and be converted to pyruvate. But there is regulation, downstream regulation of this process, but there will not be going back to fructose 6-phosphate. So it's important to understand what regulates this process. So if you think about it, right, fructose 1,6-bisphosphate is the phosphofructose kinase. Since this is such a highly regulated step, is going to be what is going to cause inhibition of this phosphofructose kinase. It's going to be ATP, right? So ATP, because ATP is the primary end molecule and the primary function of glycolysis. ATP is the presence of ATP means that that's going to allosterically inhibit PFK because we don't need any more ATP. We're saturated with ATP. And just to quickly take a detour, saturation of ATP is also called the energy charge of the cell. Okay, so energy charge of the cell, right? What is that? That is the ATP, total ATP divided by the AMP of the cell, okay? Or the total, what is the ATP ratio? Or this is roughly what the formula is here. Okay, so basically when we have a lot more ATP versus AMP, the energy shard of the cell is going to be high, and that's a way of kind of numerically identifying or numerically predicting or bucketing the progression of phosphofructose kinase activity, right? So phosphofructose kinase, just to note, is inhibited by the presence of ATP, but it's also stimulated by the presence of AMP. Now, AMP stimulation is very important. I'll get to that in a second, but more AMP will be present when the ATP is low in a cell, right? Because ATP gets converted to AMP through several processes, ADP, and then finally to AMP through several processes. And when AMP is high, then the cell automatically believes the ATP is low, and thus that will stimulate PFK1 to continue the creation of fructose 1,6-bisphosphate from fructose 6-phosphate. Okay, so this is very important to realize, because this is, again, we need to regulate the catabolic activity of the glucose in the blood, right, or in the cells. So now, one thing before we continue on with regulation steps in the phosphofructose kinase PFK1 is I want to talk about adenylate kinase. So in exercise physiology, this is talked about quite a bit, and this is actually not too complicated in its initial stages to discuss, but let's say you have a lot of AMP in the cell. We were just talking about high AMP conditions, which would stimulate phosphofructose kinase. Well, in an alternative parallel or pathway, let's say your ATP was still low and you needed ATP really quickly. This would likely occur in muscle cells or cells that consume ATP vigorously. So you can actually take two ADP molecules, right? You see this ADP here? You can take two ADP molecules and you can convert ADP to ATP by an enzyme called adenylate kinase. Now, adenylate kinase it's like a quick way of generating ATP, but what it will do, it will increase ATP, right? So adenylate kinase is it's known as a metabolic monitor, okay? Because once it releases AMP or creates AMP after it's combined to ADPs through phosphorylation. Remember, adenylate kinase means phosphorylation, so it can remember ATP is adenylyl triphosphate. Adenylate kinase works on ATP, so it also works on ADP. Adenylate diphosphate works on ADP, so it will transfer one phosphate to from one ADP to another ADP, creating an ATP and AMP. The AMP levels increase, and when the AMP levels increase, then you have these you have these AMP sensors. And then the AMP sensors, the most important AMP sensor here is called AMPK, right? And AMPK is has a long name, but it's called AMP activated protein kinase. Again, we have another kinase, right? So, and what's interesting to note, it's a highly conserved sensor of low intracellular ATP levels that is rapidly activated after nearly all mitochondrial stresses, even those that do not disrupt the mitochondrial membrane potential. So basically, it's like when AMP is at its maximal, then the AMP activated protein kinase is going to essentially sense this AMP. And this AMP protein kinase, remember how I said I would connect this with mTOR? This actually inhibits mTOR activity. Now, I'm not getting too much into mTOR. I don't even have something up here for it, but mTOR is one of the cellular or biological growth factors, and it's very important when we're considering aging, because mTOR activation is implicated in a number of aging-related pathways and aging-related stresses and things like that. And it's important for the growth of muscle cells, right? So more mTOR being active means higher muscle growth, and that's essentially, if you're a bodybuilder or something like that, you want to excite or accelerate mTOR activity, right? So you wouldn't want AMP activated protein kinase to be overactive in those cases. So I'll get to that at some other point, but I just wanted to make that tie-in here. So adenylate kinase, let's go back to that. So again, that's just in the context of our discussion of energy charge in the cells. We'll just say that this is going to actually temporarily burst the energy charge of the cell because ATP will get created. And also AMP will get created as well. So, I mean, you're using your already present reagents. So, it won't be very much, there won't be a very huge noticeable change, but that's essentially what we want to do. Now we go to phosphofructose kinase. We're going back to that, the highly committed step in the cells, right? Remember in muscle cells, okay, this is one, and in all cells, glycolysis inhibition is going to lead essentially to a buildup of glucose in the cells, right? Because glucose is coming in, it goes through glycolysis. If glycolysis gets stopped here, what's going to happen is fructose phosphate will interconvert back to glucose 6-phosphate. Notice this reversible equilibrium reaction. And then glucose 6-phosphate will essentially, you see glucose being converted to glucose 6-phosphate will not go back to glucose, so there'll be a buildup of glucose, and therefore the cells, through their blood transporters, will stop accepting glucose, right? And then therefore your blood glucose levels, because this is where the blood would be your essentially extracellular space of the cells, the glucose will increase because the concentration gradient will be balanced, and there won't be any facilitated diffusion into the cell through the blood transporter of glucose. So what essentially happens is that the cell stops glycolysis. And then another thing to note, glycolysis can be stopped in other places in the cell. Glycolysis can also be stopped besides step 3. Step 1 can be stopped at step 10, which is the pyruvate kinase activation. And so that's right before pyruvate is created. You have phosphoenol pyruvate. All right, that's the product of the 9th step of glycolysis. Pyruvate kinase will convert that phosphoenol pyruvate into pyruvate, and if that is also inhibited, the activity of that is inhibited by glycolysis. So, inhibited by the presence of ATP as well, right? So, and final thing I want to mention, because I just like mentioning a bunch of things. One thing that might not be so obvious is that when we think of glycolysis, we think of ATP generation, right? So ATP generation is going to, ATP generation is one byproduct or one product of glycolysis, but glycolysis also contributes to amino acid production too, and that's not as often talked about, but we have essentially amino acids like alanine get produced. So phosphofructose kinase is also allosterically inhibited by alanine, right? Now we'll have to, alanine is this amino acid here, and alanine inhibition of a pyruvate kinase is inhibited by alanine. So basically, if there's too much alanine, the glycolysis will also stop here. Okay, so this is another regular committed step, because after pyruvate is created, then all the downstream processes of pyruvate go on. Alanine's presence, if there's high alanine in your system, will shunt glycolysis and prevent it from creating pyruvate. And there could be a number of conditions as a result of that. So at some point we'll talk about the allosteric regulation of pyruvate, and these are what I was talking before, but that was just a little bit of a detour. So I think today talked about a lot of things, and tomorrow I will talk about the liver, yum, and glycolysis regulation in the liver. This is glycolysis regulation in skeletal muscle cells and in general, but yeah, different topic. So if you like this video, like, subscribe, comment, etc., or just watch, or hate watch. Either way, just engage. Okay, I'll see you later. Bye.